see the screen now?
Yes.
Okay, cool. All right. Neuroengineering. So I'm assuming many of you are here to learn about what is neuroengineering, what do we do, and I'm going to give you an example of one project, which is my project, and show you, okay, this is what we did in this PhD project, so you have a kind of an understanding. And Joe is going to talk more. He's going to talk about his projects too, and a little more in the breadth of it. But my basic understanding is you have some brain disorders, you want to study the brain to eventually improve functionality, and you do engineering to improve the brain. That's a very broad way of putting it. There are so many types of brain disorders that neuroscientists are studying, and I am in particular going to talk about language disorders and how are we addressing that. So whenever you have the brain, you want to study the brain, you want data. And there are different ways of acquiring data. fMRI is a commonly done technique. EEG, you might have seen it, electrodes on the scalp. And ECOG is actually electrodes inside the scalp. It's on the surface of the brain, and it's invasive. It's done for surgical purposes, for clinical purposes, but then we have electrodes and we get very high quality data. So typically in our group, at least in Benham's group, what we do is we start with data. So we start with either spiking data from monkeys or rats, or we go with ECOG data or EEG data, and then we do a bunch of signal processing on it. It could be different types of things, which I won't worry about right now. But if the goal is we want to infer something about the neural data itself, we want to say something about, this guy has this disease and we can see it because this is the problem in the brain. So we want to say something. I just I think Joe will talk more about resolutions and all these different types of data. I just want to highlight that the data that I'm using in this project is bang in the middle, ECOG. It's got very good spatial resolution, which means if I put an electrode on the surface of my brain, which is EEG, which is on my scalp, it's contaminated by my muscle movements and eye movements. So the EEG can be contaminated. ECOG is far more precise. You know exactly what location you're getting it from. Time is, it's got millisecond resolution. It's a good thing. It's not like the single neuron spikes where you get amazing spatial resolution, but not much time. No, it's very fast in time, but then you don't have any, you can't cover the whole brain. You just get one or few neurons. So ECOG covers like a big part of the brain, but it's kind of a broad signal. But anyway, I'm going to get into my project. So we all speak, we all use language, we humans are evolved to have very superior language with lexicon and grammar. But unfortunately, a lot of people have language disorders, and it's caused by something called lesions in the brain. And you might have heard of stroke. The very common problem is people get a stroke, a part of their brain dies, and then they suddenly have speech problems. Speech is just one, it could be different kinds. I'm going to give you an example. So it's been shown in 1861, that's one of the first aphasias, called a stroke in this green region of the brain causes something called Broca's aphasia, where the person is able to talk very slowly, but they have meaning. They understand and they say intelligent things, but they're just unable to pronounce the words, enunciate the words. On the other hand, a lesion in the blue region of the brain, such a person talks beautifully, smoothly, but utter garbage. I'm going to show you an example of these two aphasias, just so you get a sense of, okay, what's this research eventually? Not immediately, maybe down the line, eventually, what kind of people do we hope to help? So here's a video of a guy with Broca's aphasia.
Can you tell us your name?
Mike Caputo.
And Mike, when was your stroke?
I was 7 years ago.
Okay.
And
what did you used to do?
well, worked on a desk, seven sales and worldwide, and very good. Yeah.
Okay. And who are you looking at over there when you turn
That's
your head?
my wife.
Okay. And why is she helping you to talk?
She's a speech.
So you have trouble with your speech.
Yeah.
And what's that called?
Aphasia.
All right.
All right. Did you guys understand that? So that you could see that he was struggling to kind of speak, but he was making sense. He was able to have a conversation, but he was struggling. Now here's another kind of aphasia called Wernicke's aphasia, and completely different problem.
Hi, Byron. How are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talk with the people for them over there. They're diving for them at the moment. But they'll save in the moment. He'll have water very soon for him. With luck for him.
So we're on a cruise and we're about to
We
get to
will
Juneau.
store it right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment, they don't show a darn thing.
With the iPad that we were doing. Like here?
I'd like my change for me and change hands for me with Happy. I would talk with Donna sometimes. We're out with them. Other people are working with them. I'm very happy with them.
Good.
This girl was fairly good.
Okay, so did you guys see the difference in the Wernicke's aphasia, where he is speaking pretty fluently but utter gibberish? So back in the day, people started thinking that maybe the Wernicke's region is where language comprehension occurs. That's where you understand language. And then sort of information goes from the comprehension region to this Broca's region. And that's where your whatever you want to say is made into articulatory sounds. So it's like, oh, you have to say the word apple, you say apple, and then that information is sent to the motor region, and there it moves your tongue muscles and you speak. So they used to believe that language happened in this sequential manner, and that's an old model and it doesn't really work that way. So there are millions of people with language disorders. These are just two examples. The real problem is the many symptoms, many types of lesions, and you can't, with lesion analysis, you don't really know whether you've broken the system or is it a local problem that you have destroyed. We don't really know how language is really organized in the brain. So how can we ever help these people? Maybe in the future we want to build some kind of device that can say, okay, these regions of the brain need more stimulation so that they could sort of use plasticity, or maybe we can try and decode their speech so they can have some help. There's nothing we can do if we don't even understand the brain. So this problem is to like understand language. And the question that I'm getting into is, okay, language has historically been considered localized, which means, just like I said, comprehension happens first, and then maybe finding the word, and then articulation, then motor responses. Is it really that way? Sequentially cognitive process? Or is it a network? So the very fact that you're comprehending is a network, is a brain region, a network of brain regions working, and then a network of brain regions doing the second thing. So these are the two different types of scenarios that could be happening in the brain, and we want to evaluate which one is true. So the gory picture, but we get data from ECOG, which is electrocorticography, electrodes obtained from epileptic patients who have gone to the surgeon because they have to cut out a piece of the brain to help their seizures. But before they do that, they put a bunch of electrodes to localize the seizure, and they have to be there with 10 days with these electrodes. So they do a bunch of tasks, language tasks. And we get data from that, and our group collaborated with them. And my job was, here's this data. What can we tell about the brain? What can we tell about language? So here's a task that the patients did. The task was at time 0, they were shown that picture, not just this one picture, they had many pictures. And if you saw it, you might look at it and say, alligator. And whenever you said it, that would be your articulation time. And that's one trial. Now imagine you did this 200-300 times. That's my data set for a single patient. So I'm going to get a bit more technical now. What we believe, what we have seen is that if a signal is composed of many frequency components. It's got these very slow components, which are like delta, they move really slow. And then the really fast moving components of your signal is high gamma in the brain. So if you look at the high gamma power, you can actually tell a lot about what's going on in the brain. So I'm going to show you that. I'm going to show you what we saw from our data for this experiment. So this plot it might look a lot. This is a bunch. So, okay. So if you see the x-axis, this is time. Your 0 is when the image was shown on the screen. And red indicates that there's an increase in power in one single electrode. And these different rows are trials. So I'm just stacking the power from all these trials, one on top of the other, just so you can see what it looks like. And you can see that this is a visual electrode. So as soon as the image came on the screen, there was power in your visual cortex. Boom, high gamma power goes up. And so you can see that the power increase for all trials is aligned with the stimulus. It's about 200 milliseconds after stimulus. Now, I've aligned these trials in ascending order of articulation. So what it means is the first few trials, the guy spoke pretty quickly. The last few trials, he's like, I have no idea what this is. It took a long time, and then said apple or alligator. So it's that I've just ordered these trials. So now if I show you a different electrode, this is an electrode from the motor region where the tongue and the muscles of the mouth are moving. So the guy is articulating, and you can see the power in the motor region. You can see that that increase in power coincides exactly with the articulation time. So if I can convince you that high gamma power truly corresponds with the task, it tells you some information about what's going on in the brain. So I'm going to show you this as this is one of the patients that I looked at. The dots you see are electrodes on the surface of the brain. And the color of the dot indicates the increase in power of the brain, of that high gamma power that I talked about. So the first brain is how is the power right after the image was shown? And you can see there's a lot of red at the back of the brain. That's like the visual region. And then the second window is right before he was speaking. You can see there's a lot of power in this Broca's region, in the front of the brain, in the motor region. And in the third window, if you see there's a power increase on the auditory cortex, there's 3 triangular deep red dots kind of in the middle. That's actually the patient hearing himself speak. So his auditory cortex is having a power increase. So if you notice this way, this gamma power has this wave-like effect through the brain. And as a certain brain region is kind of participating in the main task, it has a high power. The problem with power analysis is we have no idea what's going on underneath. If there is an increase in power in some regions, there's a decrease in power in other regions. We don't know whether information was sent to that region from which region. Do we assume that information went linearly? We don't really know. So going back to the original question, which we wanted to know is, our brain regions, cognitive processes, are they local or distributed? So this power is really what we mean by well localized. So if you see the analysis here, the visual region has high power, so the visual region is active. Is that the only way? Is that the only region active? Maybe some other region without power might be active too. We just don't know. So this view of power assumes that local activity is the most important thing. But my question is, what about network activity? So let's see how we address that. So this is where we get into kind of the heart of the research. What we decided was we took this time window from when the person saw the image to when he spoke, and we divided that into smaller time windows. And in each time window, we said, let's find out how much information is going between every pair of electrodes. So if you see between X and Y, you can get 1 point in this matrix. So if you imagine, I have 100 electrodes, I have 100 by 100 matrix. And a red point in this matrix means, oh, between electrode 81 to 10, there was a lot of information flow in that small time window. So what we did is we took this task. So if you notice, the I is when the image was shown, and articulation is somewhere in the middle. I have 40 windows now, and I have a matrix or a graph, what we call a graph, at every time point. So you can imagine that each of these graphs correspond to some kind of signal on the brain. So it looks like a matrix, but it has a representation on the brain. Every node is a number. So the 10th row, for example, could be one of the nodes here. So it's, if you don't understand right now, you got to tell me, because I have no idea.
I have one quick question.
Yes.
Have you done any counter analysis of a normal human brain to compare where the brain activity is?
You cannot do normal human brain with ECOG, because ECOG is by definition invasive, and normal people are not going to cut it up. But yes, EEG, you have done shows very similar patterns of activation. Even though the high gamma is actually attenuated, you have lower frequencies. Moreover, we chose these people, even though they have epilepsy, to be as language normal as possible. So there were language tests done that language was on the left side of the brain, and they had sort of normal language as compared to regular people. So it's as close as we can get.
Okay, I was just wondering for a comparative analysis of if there was any difference in where the gamma rays were most prevalent.
Yeah, well, I've actually seen there are slight differences between people, but on a broad scale, from other analysis like EEG and whole bunch of work from monkeys, where they do invasively go on healthy monkeys, they're around the similar regions. At least
Okay,
the big areas, they kind of, you see that.
thank you.
No problem. So what I'm now going to tell you is, we have these scale of graphs, and we want to extract network features. So kind of looked into this, where at a very broad scale, let's look at, for every scale, give me how many connections do I have in every time window? What's the connection density? At a more intermediate scale, what are the communities? Are there any clusters in the brain regions? At a finer scale, I look at like in degrees, out degrees. There's something called coreness. So I'm not going to get into the details of this because I don't want to overwhelm you guys. But I'm just going to show you like a high level. So coreness of nodes is like, how central is that node to the network? So the red nodes are really central. So it's got a higher coreness. Communities is like, how do you group the nodes or your network into little groups that you find separable groups by a definition, where you say the connectivity inside one of the groups is greater than connectivity between the groups. That could give you some information. And then we also did in degrees and out degrees. So remember, I can do the node level analysis. I can do this for every patient and for every node. So I have all these bunch of node metrics. I also have a bunch of power metrics. How much was the power in this time window compared to baseline? So I have all these different metrics, and now I can start comparing. So I just wanted to show you, this is maybe visually easy to understand. This is what we get from the Louvain analysis. So literally, this is from one of the patients. In the window before articulation, you actually see you see the red cluster is actually right over Broca's region, connecting a bit into the motor areas. And the blue cluster is pretty much frontal temporal, some of the frontal regions, which are known to be having some relation to figuring out articulation. and a whole bunch of motor and auditory cortex. You can see that after articulation, you actually have the red cluster now is a lot more on the auditory cortex, which basically shows, okay, you can actually see these different graphs smoothly change, showing these brain dynamics. Just wanted to show you that these are complex graphs, and they also vary from patient to patient because it's an individual level that we are extracting this. So how can we now answer this question? Does power give you more information about the brain, or do network features give you more information about the brain? So we came till here, and then we did a validation study. So this might be like your control that you were talking about, but we use the same patients because these are so dependent, patient to patient, they vary. So what we did was the gold standard, the current gold standard for figuring out where is language in the brain is literally in the neurosurgeons in the surgery room. And when there are, if you remember, I told you these are epileptic patients and they're going to cut a piece of their brain out because they have too much epilepsy. They figure out where is epilepsy originating, and they remove that part of the brain to hopefully solve the problem. But they don't want to cut out language parts of the brain, because you cut out the wrong part of the brain, or you cut out too much of the brain and they can never speak again, and it's a problem, as you can imagine. So this is the gold standard. They take a pair of electrodes and they zap it with current. This is like high current. It's called cortical stimulation direct. They go zap. And when they zap it, they make a temporary aphasia and in that region. Well, if that region causes an aphasia, you say, that region is language positive. If that region did not change any language, then you say that's language negative. So very shocking to hear that it has lots of side effects, but they're not permanent. It's the current gold standard. The side effects is sometimes they induce seizures and it can be a little uncomfortable. But anyway, that's what they do. So for these very same patients that I showed you all this data, they also did direct cortical stimulation. So I'm just showing you what that looked like. So for example, for one of the patients, I had 5, I'm calling it node pairs because I had a pair of nodes was stimulated at a time. So if you can see those blue lines in the back of the brain, it's like kind of underneath, it's called the medial surface. So those were the 5 node pairs that were positive, which means when that person was doing the language task during DCS, they actually couldn't speak or it disrupted the task. But all the yellow regions, it did not disrupt the task, so they're language negative. And for a different patient, they're in different regions. And we had 14 for another. So I had all these, I actually had 4 patients, bunch of positive and negative. And here's what we did. We did a machine learning task where we said, let's see if we can classify, we know the ground truth. We know if these node pairs are positive for language or negative for language. Let's see if we can now classify them. We use a little for training, but then we test. And what's our accuracy to divide? This is language positive, this is language negative, but we only use power features to do the classification. How good? Well, what if we use only network features to do the classification? How good do we get? So we kind of did this, and I'm just showing you the results because the details can get too much. So what we found is we had around, so this is average of multiple classifiers and multiple patients, and we had around 62% accuracy in identifying language positive and language negative areas using just power. But when we use power and network features, particularly coreness of a node, how core is a node in the network, that feature had like 75.9% accuracy and was significantly high, which you'll get into significance when you're writing a paper. So we finally got our paper in Neuro, so I'm happy about that. So yes, I was trying to see if oh my gosh, I'm already out of time. Okay, I'm done though. So what we find is, in conclusion, we find that the evidence points to distributed networks. We believe, and most of the latest research is kind of pointing the same way. Language is not localized in the brain. It's a network of brain regions doing a task. another network of brain regions doing a task. And when you have a lesion, you've kind of broken that network. And depending on what exactly in the network you broke, you have a different kind of aphasia phenomenon, aphasia type of aphasia that is manifest. And that's our group. And thank you. So yeah, if you have questions.
Sudha, there was one question from Loretta about plasticity.
Oh, good.
Yeah. I don't know if you can see it in the chat. It was a few messages ago.
Have you stopped the share? Okay, awesome. If brains have plasticity, shouldn't other areas of their brain, like cerebral cortex? Yeah, great question. It's the way the brain works is you can't have plasticity between completely disparate regions. Generally, the area close to where you are affected tends to take over the most. Or sometimes, if your left hemisphere lost some functionality, sometimes right hemisphere will try to take over. Sometimes it's again one of those things it's observed is sometimes people are able to regain their functionality with time, but sometimes people cannot regain it even after decades. So it's not very clearly understood. Maybe you need to have some amount of functionality left so plasticity is able to help you. But our the idea is you can have brain machine interfaces that are doing really cool things. We can't do that until we have awareness of where to use these interfaces. So yeah. Any other questions before Joe takes it over? Of course. Right, Joe, I think it's yours.
All right. Let me go ahead and make sure. Okay. You all can see the slides?
Yes.
Excellent. Cool. Great talk, Sudha, as always. So I'm Joe. Sudha and I are both, well, I guess I'm formerly technically PhD students in the same lab. And so that's Dr. Behnam Ajang's lab. He actually heads this whole initiative that you see in the corner here, which is the Rice Neuroengineering Initiative. Super cool. I'll talk a little bit about that here. But overall, I'm trying to give you a sense of what neuroengineering is, how we think about it, particularly at Rice. And so there's some different areas you could think of, like restoration. So trying to basically restore sensory perception that people should or used to have, and those kinds of things. There's also, reproducing different things, say in AI and robots. And there's also enhancement. You could think of the Minority Report type thing. And so we actually have faculty that, well, let's see. I think we especially focus probably on the first and the third areas at Rice. And so really the goal, one of the outcomes of this, one of the products is that you better understand the brain by trying to do all of these things. And so there's different challenges that you run into when studying the brain. The one on the end there, nonlinearity, that's probably the one that Sudha and I think about the most. And what that means is there's all these assumptions that you could make that would make it really easy to study data. So if your system is linear, if it's Gaussian, which Gaussian is a fancy way of talking about bell curves. We often talk about grades being distributed in a bell curve. And so the data from the brain does not follow these nice assumptions. And that actually says a lot about the current state of neuroscience. People use tools all the time that make these assumptions, linear, Gaussian. And so a lot of our focus in our lab has been trying to develop methods that don't make these assumptions, because the brain breaks all of these beautiful things that we wish we could assume. And so that's why it's so great to study. And you always see this thing about the scale of the brain. I mean, it's worth reiterating because it's always so insane. There's 100 billion neurons in the whole body, 21 billion neurons in the cortex alone, which is the gray matter. And then there's some insane number of synapses. I've seen different estimates. It's never clear to me, but 150 trillion is probably a reasonable number. And yeah, a lot of my studies have focused on the cortex. It turns out there's a lot of interesting things just even on the outer part of the brain. I think a lot of times we think, oh, you have to go deep in. You do sometimes, just depends. And of course, even the neurons themselves are quite complex. I think something I didn't know for most of my adult life is that there are different types of neurons. And so this causes a lot of problems for experimentalists. In the cortex, you would look at pyramidal cells a lot. That's the common type there. But then, we talk about motor neurons for different degenerative diseases. And so it's just, I hope I'm compelling you that neuroscience is very interesting. Now, electrical engineers, we get excited because there's so much electrical activity in the brain. And if you wanted to take a super simple approach to a neuron, you could think of it as a battery. And so if you measured the voltage across a battery, well, you'd get 1.5 volts if it's a AA battery. And similarly, believe it or not, you could look at the inside of a neuron and the outside, like the surrounding area of it, and get a voltage difference. And so if we measured that, you would get negative 70 millivolts. And so basically the inside of the neuron is actually at a lower voltage than the outside. And that's at a so-called resting state. So it's when the neuron's not doing anything, it's just hanging out. And so we call this the membrane potential. It'd really be the resting membrane potential. And so it'll sit there, but then you start to get activity from other neurons, which would be EPSPs. Not really going to go into that right now, but it's activity coming from synapses, connections of other neurons. And so as other neurons fire and they're connected with this neuron starts to get a little excited. And then once there's enough input from other neurons, it gets super pumped and it spikes, which would be called an action potential. And basically the actual voltage that we've talked about, it reverses. And so the inside of the neuron is actually at a higher voltage than the outside. And so we care about these spikes. These are our obsession. It's pretty much thought that spikes contain all of the information, particularly the timing of a spike. So when does a neuron fire, especially relative to other neurons firing? And so, yeah, I've talked about that a little bit. There's a threshold, I guess, negative 55, depending on the neuron. And so the way that we analyze this data often is we look at what we call raster plots, which are really spike trains, so just spikes in a row. And so what you would do is, let's say you had one trial of an experiment, you're recording from the brain of a monkey. Well, over time, you could just plot vertical lines to indicate that the neuron you're looking at fired at that particular time. And so you'd keep on doing this and you'd get all the times it fired. You could look at more trials. And basically you would get this cascade of spikes. And so this is our raster plot that we look at. Now, this actually has relevance because here's recordings from an actual monkey, the visual cortex of a monkey. And what was done is first the monkey was fixating on a dot, and then a target image, which just means that a literal picture on a screen was shown to the monkey. And then that picture disappears. There's a 1 second blank period. And then the same image appears for, I forgot to mention, both image appearances are for 0.3 seconds. So when the second image appears, it's possibly rotated and then it disappears and the monkey is holding onto a lever. It's supposed to let go or keep holding on, depending on the rotation. So they actually get these monkeys to learn how to do a task, and they incentivize them with orange juice, literally. They get drops if they are correct. And so what's cool is we look at this data and you can see that when the two images were presented, we see completely different neural activity from the other times. So this neuron, for example, was firing really heavily when nothing was being presented to the monkey, no stimulus, no picture on the screen. And then when the picture's shown, it is much more selective about its firing. And you see the same thing for when the second image is shown. And so it tells us that there must be information here. I mean, there's something going on. This is describing the external world. And so it's just really cool that you can actually visually see this going on. Now, Rice is a little interested in this kind of thing. The Neuroengineering Initiative is a $49.5 million investment. I don't know why they didn't just round it to 50, but I'm not in charge. So yeah, so they're very interested, and we have a lot of faculty who work on this. Ajang is our advisor, but there's a lot of other people working on neuroengineering projects. And so actually that's Ajang in the picture, and that's me, I think. Oh yeah, Sudha, you're there too. Anyways, but yeah, so it's a really cool initiative, and I'm trying to advertise a little bit because I think all of you would be great people to start talking to us. And I know they've had high schoolers involved in research projects, I believe in the Kamiri lab. And so he's like this cool mix of experimental, but also like looking at the data and doing crazy models. So we have a lot of interesting people working on this. Now, you might think, okay, this is all super interesting. Why is now the time that these things are really developing, moving forward? Well, there's a lot of technological advances that have happened. And so one of the ones you're going to see me talk about is genetic engineering. We can actually get neurons to light up when they fire. So literally you could point a camera at the neurons and just look at the pixel intensity, like how bright is the neuron? And that'll tell you if the neuron's being really active or not. But then there's also the investment, which I talked about, Rice investing. There was the Brain Initiative. I actually had a grant related to that. And so when we're engineers looking at this, we get really excited because we're trying to restore people's senses or just motor control. And so you can see here that with this deep brain stimulation technique, they're actually able to effectively stabilize the woman's hand. And so it's amazing technology, and it changes people's lives. I mean, you can see the difference without the technology and with it. So we're incredibly excited about these things, and we really want to create solutions that can help people. And so I look at a different part. I'm looking more at sensory systems. And so the question I'm concerned about is, how do we experience the world? You know, it's a very large question. You could have philosophical answers for this, but I just look at the senses. And so these are our classic 5 senses. And so all of these are based on receptors, which are really just proteins embedded in the effectively input of neurons. So neurons have dendrites, which are these like tree root-like structures. And so, yeah, you have the proteins in those structures. There's different types of proteins or receptors. So you have photoreceptors responding to light, mechanoreceptors, which respond to pressure, sound waves. Chemoreceptors are my favorite because they're the most complex. And so you think about it, well, a receptor is trying to describe a molecule. And if you start thinking about all the ways that you can describe a molecule, you start to realize this is a very complex problem. And it's actually an unsolved problem. We understand, arguably we understand these first 2 quite well. But chemoreceptors, which underlie smell, for example, we really are at a loss. And so we need smart people like you all to come in and do neuroengineering to help figure this out. And so when we talk about sensory encoding, say for vision, what that's going to boil down to is at a certain level, it has to do with edge detection and the orientation of edges and bars. So very fundamental visual features are described by neurons in vision. And so we talk a lot about these so-called tuning curves. And basically your x-axis would be some feature of the stimulus. So in this case, you have a light bar on a screen, just a bar of light against a black background. And if you orient the bar in different ways, the neuron will fire at different rates. And so that's what this y-axis is. It's basically the activity of the neuron. And so in this cartoon example, the neuron is super tuned to a perfectly vertical bar. And so any different orientation, the neuron fires less. So we talk about neural tuning a lot, and that's going to be a mystery when we get to olfaction, sense of smell. And so, but before we get there, it's just worth mentioning for audition, our sense of hearing. This is like a pretty intuitive system where the neurons are describing frequencies. So just, you have somebody speaking, there's different frequencies of sound in that speech, and the neurons will be tuned for particular frequencies. Now, I've been hinting at olfaction, which is the one I study. Again, sense of smell. Sometimes people don't know that word. And so there's 2 early structures that are involved in your sense of olfaction. And so both across rodents and humans, they're called the olfactory epithelium and the olfactory bulb. And so I'll zoom in on the rodent one just to show you what we're talking about. So on the right-hand side, you have your odorants, which are just molecules. And actually just anything you can think of, it has a chemical structure, carbon, oxygen, et cetera. These are floating in the air, and they are interacting with the dendrites, the inputs of neurons. So those proteins we've been talking about, the receptors, chemoreceptors, they're embedded in these claw-looking dendrites of the olfactory sensory neurons. And so these OSNs, for short, they're going to respond to the odorants, and they're going to attempt to describe the odorants so that ultimately you can understand the smell, you can perceive it. And as I was hinting at, well, there's a million ways to describe a molecule. Everyone has their own lists. You look around the literature, somebody's made a list of 1,000 features to describe molecules. Another person has another 1,000. And so it's totally wild. And whatever way that these OSNs are describing odorants, it's being transmitted to sites in this structure called the olfactory bulb. And so OSNs that are of the same type, which means that they describe smells in the same way. So, you know, the red cells will describe smells in one way. The green cells will describe smells in a different way. And by describing them, I mean they fire, you know, a certain rate per odorant. And so, yeah, the same types, they go to the same spots in the olfactory bulb. We have a complicated name for those spots. It's glomeruli, glomerulus for short. And so then you have neurons, mitral cells, which will transmit that activity. They relay that activity to the cortex for further processing. And so all of the anatomy of this system is pretty well studied. We know that all of these neurons exist. Yet we don't know how the neurons are describing odorants. We really don't have a tuning curve, as it were. And so this is, you know, you could have a cartoon example. And what we're showing here is that, let's say these are all receptors for a particular, you know, sensory neuron. Well, given an odorant which has different features shown as cartoon rectangles, you know, half circles, whatever feature it could be, number of carbons, that kind of thing. And so what you'll have is that like one receptor can be activated by multiple odorants, and one odorant can activate multiple receptors. Because, and this is really confusing, because what you would like in this beautiful diagram is that, okay, the red neuron only responds to X odorant, the green neuron only responds to Y odorant, but that's not what happens at all. It's this crazy mapping of it's not a one-to-one mapping, it's a many-to-many mapping. And so you could show this in different ways. This was an experimental result where they actually displayed this. And so what they're showing is they have different olfactory sensory neurons here, and then they have different odorants that they've introduced, and the radius of the circle is how much that neuron responded to the odorant. And so what you'd see for a particular neuron is that many odorants caused responses for it. So you can see that there's many responses here. And then for a particular odorant, okay, you get different neurons responding to it. So this makes it really difficult to study. And what makes the situation even more difficult is that there's not 10 of these. There's 10 million olfactory sensory neurons. Now we're able to record from more and more neurons over time, but 10 million is not a number that we're anywhere close to. And so the way that you cheat is you study glomeruli. You look at these zones where the neurons are you know, targeting, that they're going to. And so there's only 3,600 of those. And in fact, when you consider that there's duplicates, you can get the number down to about 1,000 unique glomeruli. And so it's really a much easier system to study. And we effectively can use it as a proxy for studying the activity of these olfactory sensory neurons. And so something that I focus on is not only this question of how are those, this, by the way, is a more abstracted diagram of what we just saw. So at the top, you have the odorants interacting with the OSNs, then you have the glomeruli, then you have mitral cells transmitting. So more abstract. But yeah, so we have this one question of how do the olfactory sensory neurons describe odorants? And then this other question that I'm looking at as well is the role of these cells called SSA cells, superficial short axon cells. They connect glomeruli. We know that they connect glomeruli. We've seen them before, but we have no idea like anything beyond their existence. We don't know, are glomeruli that are close together? Do they have stronger connections? Is there some spatial layout to this connectivity? So that's the second question that I've been looking at in my research. And so, yeah, it would be, what does interglomerular connectivity look like? And so this is complicated because it turns out that olfactory sensory neurons they don't only respond to odorants, they actually respond to just the sheer act of breathing. So just by the mechanical stimulation of breathing, they respond. It turns out that and so there is some controversy here. Some people say, oh, it's odorants that weren't filtered out in the air that you're using. So I'll note that. But there seems to be evidence that it could be mechanosensation, meaning that there's not only chemoreceptors in these neurons, but there's also mechanoreceptors responding to the stimulation of breathing. So you could do an analysis where you effectively get a breathing time series just by looking at the change in temperature around the nose. That'll tell you basically the rate of breathing, a breathing time series. We do this crazy genetically, we do some genetic alteration in order to make the neurons light up. We'd call it a genetically encoded calcium indicator. So basically the neurons will fluctuate in fluorescence based on the activity, because the calcium activity in a cell is a proxy for the firing of a neuron. Now it's more complicated because there's a bunch of other things that modulate calcium in a cell. So there's a whole host of issues in studying this type of signal. But I would encourage you to look at that type of signal because it's increasingly common, because you can record from a lot more neurons. Since now you can just point a camera at the zone and you can see a bunch of neurons, rather than having to just put one electrode or basically a needle into the brain and getting one electrode recording. So anyways, yeah, you would, if you looked at this activity of the glomeruli, you would see that it's in sync with breathing. And so this is a problem because everything will look related. Even though they're not, it might not actually be the connections between glomeruli that's causing them to be related. It could just be that they're all responding to breathing, all responding to the same stimulus effectively. So you get into this existential question of, well, how do I know anything's connected? This has played out in really funny ways, just in the world, where people have found false relationships and they don't know how to say from the data that it's a false relationship. So, I think the best example is this chart of polio and ice cream sales. And so people were saying, oh, okay, here's this anti-polio diet, don't eat ice cream. That's how you prevent polio. And so obviously we know this whole thing is absurd. Like ice cream sales and shark attacks are not directly related. There's not some like causal relationship between them. And so the variable that you're not accounting for is seasonality. And so when we're doing this analysis of connectivity between glomeruli, the variable that we need to account for is breathing. We need to adjust our analysis for the activity of breathing. And so you can make diagrams for this, make it more abstract. And you get this notion of direct and indirect connectivity. And this is all really important when you're studying the brain, because you don't want to say that two brain regions are directly connected when they're actually indirectly connected. They're modified by some other stimulus. And so we worry about this all the time. We have different names for the configurations that can cause this. And so in general, what you do is you account for the activity of other nodes, which could be brain regions. So when you're looking to see if brain region X is connected to brain region Z, what you would do is you would condition on another brain region. So you effectively would look at the data for this brain region Y when you're looking at the connection between X and Z. And so in this toy example, let's say that there's no unique influence on Z from X when we account for Y. And you would basically eliminate that edge, and we'd call it a direct graph. So this is something we think about as well. This is really helpful for the glomerular analysis. And I often do this kind of analysis in the so-called frequency domain, which is what Sudha was talking about when she talked about different frequencies of activity. Just to underscore that these frequencies matter, while we talk about brain rhythms, I think these are especially popular like in society. People say, oh, is something wrong with your alpha rhythms or that kind of stuff. But these rhythms do actually have meaning. They've been shown to be associated with different behavior. So you could get these recordings just from EEG, just putting electrodes on the scalp. And so these activities matter, they're interesting. What I do is I consider a recording from one side of the brain, just one part, one electrode. And you can decompose that recording into these frequencies. You can see, okay, how much low frequency activity do I have in the recording for this site? How much high frequency activity? And then you can consider other sites. And most interestingly, you can consider the synchronization of the rhythms between these sites. And so you can be asking questions like, how in sync are the gamma waves over here with the alpha waves over here? And so we'd call this frequency coupling. It's just one term for it. And so it's really the synchronization of brain rhythms. Now, there's different scales of activity that this can be studied at. Spikes underlie everything. And so there's a really complicated method to get individual spikes, which is called a patch clamp. You can't do it at scale because basically you put like a pipette against a neuron and then you suck a piece of the skin out. And that allows you to to actually record from the inside of the neuron. And so since you also have a recording from the outside, you can get that membrane potential. That's how a patch clamp works. And people have actually made robotics to do larger scale patch clamping, like automated pipette goes to neuron, sucks the skin out. All this crazy stuff. So anyways, I diverge and I know time is running out. So I just say this to mean that there's different scales for which you can study this synchronization between brain rhythms. And there's different thoughts about what this activity means. Perhaps neurons use solely their timing to indicate when information about the outside world. Perhaps it's just the number of spikes over a given time. Maybe there's some background oscillation like the breathing I was talking about. And so their neurons will fire at a particular phase in relation to that breathing cycle. So I just want to throw out this notion of coding because it's really interesting and it's definitely a neuroengineering task. But anyways, coming back to the olfactory bulb to kind of close this up. Well, I showed you that there were different scales and ways to record neural activity. I've been hinting at this genetically encoded calcium indicator technique. And so that's actually what you do for the olfactory bulb because the bulb is fairly accessible in terms of surgery. You don't have to go too deep or anything. And so that makes it easy to tear off some skin, politely with respect to the rodent, and then put a camera there and record from these glomeruli as a proxy for the activity of the olfactory sensory neurons, which we want to know more about. And we also want to know about this connectivity between glomeruli. And so we have this crazy setup. Well, actually, sorry, we don't have it. Our collaborator across the street over at UT Health has it. And that's another cool thing. If you do neuroengineering at Rice, there's a lot of collaboration going on with people in the Med Center who are experimental geniuses. And so they have this crazy microscope setup that allows them to look at the calcium activity of these neurons, just camera looking at fluorescence from glomeruli. And so if you were to actually do this, what you'd have is, well, first, we just have kind of a full frame view of what the camera is seeing in the olfactory bulb. And so these ellipses are the contours are indicating glomeruli. Might not be immediately clear in this picture. They would have different ways of determining how big the glomeruli are, where they are, etc. Anyways, what they do is they select a few pixels within those contours, and that gets them effectively glomerular time series. And so they could just look at the pixel intensity, the brightness over time, and that would tell them how active the glomerulus is. And so this is really cool when you introduce some odorants. So here we're introducing 3-CHO, and you can see at glomerulus 5, no response, doesn't care. Glomerulus 4 has a really strong response. Then you consider a different odorant, 4-CHO. Well, glomerulus 4 responds in a weaker fashion to that one, whereas glomerulus 5 all of a sudden very active. And so you can look at these different types of behavior, different responses to odorants. And this is how we ultimately hope to solve this problem of, what features of odorants are olfactory sensory neurons responding to. So we're using, again, glomeruli as a proxy for the activity of those neurons. Now, you also have spontaneous activity where there's no odorants introduced. And this is the kind of activity where you would see coupling to breathing. And that's actually what you're seeing here with these oscillations. So it's the glomeruli responding to breathing. And so if you were try to correlate, just look at a relationship between glomeruli, well, you're going to get a bunch of relationships just because they're responding to breathing. It's not that they're actually necessarily connected or related. And so this just further underlies that point of, okay, if we consider spontaneous activity, what can we find? I'll skip this for now. And without really going into detail on these acronyms, because this is a slide for my defense that I didn't get to simplify. Anyways, what you're seeing here is you're seeing different levels of frequency coupling. So different levels of relationships between glomeruli. And we're very suspicious that these relationships are due to just the glomeruli responding to breathing. And if you actually do an analysis where you condition on breathing, you adjust for breathing, you're not just looking at shark attacks and ice cream, you condition on seasons, you remove the effective seasons. Well, we get basically no relationships between the glomeruli. And so this tells us that we need to be very careful when we're studying relationships in the brain, because we might get a bunch of false indirect connections. And so that's the main result there. The thing I didn't get to tell you too much about today is how we're studying how we're looking at olfactory sensory neurons and what they describe about molecules. And that will boil down to actually a machine learning problem, talking about feature space and trying to predict responses of glomeruli to odorants. So if you're ever bored, you can email me, probably get my contact through Mahmood, and ask me about that, or any questions about the Neuroengineering Initiative. I'd love to talk more about it. So that completes my talk. Thanks for listening.
All right. Thank you to our presenters, of course, and to everyone who came. I know we went over time a little, but I'm gonna drop the survey in the chat before we start our Q&A for anyone who has more questions and wants to stay and ask questions. For our survey, we will be doing a giveaway to those who fill it out, and we will be sending everyone who chooses yes for the question, if they want a small care package to be sent to them for attending the first webinar, we will do so. And yeah, before everyone goes, I would love if I could take like a screenshot of everyone. If you're comfortable to come on camera, that's fine. If not, just maybe do a little reaction, and that would be perfect. So let me drop the survey in the chat, which I will also email out to those who pre-registered in case you don't get to do it as of now. And then we'll take our photo and start the Q&A for those who wish to attend.
Oh, can I comment on the question for the Neuroengineering Initiative?
Oh, definitely, go ahead.
Awesome. Yeah, so it was asked about, is this initiative part of a graduate program at Rice? It's really interesting because, yeah, it spans multiple departments. Sudha and I are electrical and computer engineers. Our advisor is in the same department, electrical and computer engineering. And there are faculty that are part of the initiative that are also in mechanical engineering, bioengineering. I think we probably have people involved who are in computer science, maybe. But I mean, it spans multiple departments. And so if you go to the website, it'll list out the faculty and you could kind of look at their associations. But yeah, you wouldn't strictly get a PhD in neuroengineering. It would be in whatever department you get, but you would be doing stuff that's part of the Neuroengineering Initiative. And that comes with a lot of resources, like they have a ton of lab space in the BRC, which is our biosciences research collaborative building. So it's super fancy, modern, very cool. And you probably could get a tour if you emailed someone, if you wanted to check out lab space at Rice. Well, a tour, I don't know exactly what it looks like right now in COVID times, but maybe it's possible. I don't know.
All right, so I'm gonna screenshot the screen real quick so everyone can like thumbs up or peace sign or whatever, or if you have your camera off, maybe do a reaction, whatever you're comfortable with, it's all good. All right, in 3, 2, 1. Okay, I took like 3 pictures. We should be good. Thank you to everyone who came. Like I said, we will start the Q&A session after this for anyone who wants to ask more questions. But if you need to log off, that's totally fine. If you didn't get to fill out the survey or didn't catch the link in the chat, I'm going to email it out once again to everyone who pre-registered, and we'll make more announcements about the giveaway. And yeah, just thank you for coming. It was a wonderful experience. Thank you to our speakers who were able to discuss about their research and everything. And thank you to all of my friends and to new faces that I haven't seen before who decided to come and support this initiative.